I would like to start my presentation entitled Water Movement in Alpha Point Knockout Mouse Brain After MCA Occlusion. I have no COI. This presentation is deeply related to the previous study. It's a collaborating study with Professor Yasui's lab. The paper is published in Frontiers in Neuroscience 2023. The background: the neurofluid has become the very important topic in the field of neuroscience. The pathway for water movement in the brain are still unclear, although it is thought that dementia and sleep dysfunction are related to abnormal water movement. It is likely gadolinium leakage to the brain is affected by water movement as well. Aquaporin 4, AQP4, plays an important role in the water movement throughout the brain. It is a protein embedded in the cell membrane and regulate water movement between intracellular and extracellular spaces. It is highly expressed in the end field of the astrocyte and may contribute to cell swelling in the ischemic brain. So the tracer that has a similar biodistribution to water would be very useful for the study of water movement in the brain. However, a few suitable tracers are known. In a previous study, and this is also collaborating with Professor Yasui's lab, we reported on an in vitro CARS experiment where extracellular H2O was rapidly replaced by D2O. As D2O is invisible in CARS microscopy in our setting, the dynamics of D2O can be monitored by the gradual signal loss as it exchanges into the intracellular space. Using this method, we succeeded to observe a difference in the exchange time between aquaporin 4 non-expressed cell and well-expressed cell. If we could do a similar experiments inside an MRI scanner, it might enable us to monitor the in vivo dynamics of D2O. In fact, this is possible because the exchange of H2O with D2O also induces signal loss on proton density weighted MRI. So the purpose of this study is to compare D2O distribution in wild type and aquaporin knockout mouse brain after MCA occlusion using dynamic PDWI performed after D2O intraperitoneal injection. And the method. And 5 wild-type mice and 6 aquaporin 4 knockout mice were used for this study. And middle cerebral artery MCA occlusion was performed Tamara's method 3 hours before the dynamic imaging. The dynamic proton density weighted imaging was performed 4-shot EPI with these parameters shown in the slide. And to evaluate the signal change relative to the pre-injection scan, we use a %S that is calculated using the average signal of the pre-scan as SL. We create this kind of signal change map. The %S map clearly shows distribution of D2O and that flow into the brain like this. This is the result slide. The slide shows percent S maps for wild and alkaline knockout mice. In fact, region, it's high intensity in DWI images. Initially, both type mice had a small change on percent S map. After that, the faster signal decrease for wild type was observed. Sorry, it's okay. I thought I can show the arrowhead, but nothing. But anyway, please check the infarct region. It's a clearly different signal change in between the wild type and alkaline mice. Anyway, so in ACA territory, the wild type mice had similar change on IPS and contralateral side. On the other hand, alkaline knockout mice had slower change on ipsilateral side. Okay, so it's a little bit different change on the alkaline knockout mice. Okay. And the average signal for each noise were further averaged for each mouse type. The infarct region relatively slow signal decrease were observed in both types. So it's a blue dot in center figure and right figure. In ACA territory, wild type mice had a similar rate of change on IPS and contralateral side. On the other hand, alkaline knockout mice had a slower change on ipsilateral side. It's a green dot in the right side. To quantitative evaluation, we calculate the value of the slopes in the initial slope. Original slide, I can see the calculation window, but this slide, it is difficult to see. Anyway, the initial maybe 5 minutes, we calculate the value of the slope, and the result is in right side. So the slope is significantly smaller for the wild type mice than that for alkaline knockout mice. Means D2O flow into the infarct region faster for wild type mice than for alkaline knockout mice. And maybe this is very reasonable because alkaline knockout reduces the water exchange in the brain. And to quantify the signal decrease in the ACA territory, we use the time to minimum of signal change. Type 2 minimum on ipsilateral side of alkaline knockout mice is significantly slower than the other regions. And there is a significant difference between the wild and alkaline knockout mice, even on the contralateral side. Okay, so to check the hemodynamics in the HCA territories for both mouse models, we performed a dynamic large FOV 2-photon microscopy after injection of an intravascular tracer fluorescent dye. as seen in the movie on the left side, the distribution of the dye in the vessel was delayed on the ipsilateral side. This is also clearly visualized on the time to peak map in the center figure. This result was same for both mouse model, which suggested that the CBF decrease in the ipsilateral ACA territory is purely due to the MCA occlusion and not related to the type of mice. So in this case, why was similar detail change obtained on both sides for the wild-type mice, even under such an unsymmetrical CBF condition? And this is our question. So our possible answer of this question is shown in this slide. So even under the ischemic condition, wild-type mice may maintain tissue water supply in the ACA territories by increasing the number of active aquaporin 4. However, AQP4 knockout mice, some alternative paths for tissue water supply may already be active in the normal condition, and hence there is no way to compensate for decrease in CBF in the ischemic condition. So, and finally, I would like to introduce our recent research using O17 labeled water. The title is Discrepancy in the Distribution of O17 labeled water and gadolinium DOTA during and after intrathecal infusion in chronic unilateral hyperperfusion model mice. This research is being conducted mainly by Dr. Manami Takahashi and Dr. Hiroyuki Takua. So in this study, mild unilateral chronic cerebral ischemia was made by permanent unilateral common carotid artery occlusion, UCCAO, 1 week before the MRI. Intrathecal infusion of O17 labeled water or gadolinium DOTA was performed during dynamic T2 weighted imaging or T1 weighted imaging respectively. Here are brief results. And right and left figure, all 17 label water admission group, all 17 label water were distributed mainly to the ipsilateral side. On the other hand, gadolinium DOTA administration group, gadolinium DOTA was accumulated mainly in contralateral side. So it is very interesting to see that kind of quite different distribution of O17 labeled water from that of the gadolinium DOTA. These results may provide new insight for aquaporin 4 and neurofluid research, and it is suggested that only tracers equivalent to water such as O17 labeled water and D2O can measure the true water movement in the brain. The detail of this research will be presented at ISMR M2024, so I am looking forward to having active discussion there. So in conclusion, we succeeded to observe D2O distribution in ischemic brain using dynamic PDWI. There are significant differences not only in the infarct region but also in ACA territory. Aquaporin 4 knockout affected D2O distribution, especially in the ischemic condition. That's it. Thank you very much for listening. ありがとうございます。